Okay, perfect, thank you. So yesterday we had the first ever basic science track day at the annual CCF conference. And I think based on what I've heard, it seems to have been very successful. We had remarkable speakers and moderators with diverse expertise from around the world. There was lots of enthusiasm, robust discussions, and interactions. And we've actually, there have been several collaborations that have already been set up. And so I'm grateful to Stacy and Juan for recognizing the need for a dedicated basic science program and for pairing me with a wonderful co-chair, Daniela Sia. And a huge thank you also to the CCF staff who really made the program seamless. Sydney, Allie, Tammy, and so many more all worked tirelessly to make this program possible. There have been tremendous advances in terms of technological advances such as single-cell omics, as well as novel therapeutics such as nanoparticles, cell-based therapies, which have catapulted cancer care into a new era. And these advances really served as the inspiration for our program. And the program had 4 sessions, and each session we had 4 talks with at least one talk based on general cancer biology. So the first session was Technologies and Innovations Revolutionizing Cancer Care. And we heard from Dr. Joel Schneider, who is a wonderful chemist from the National Cancer Institute, and he gave a really insightful and he explained the chemistry so well, talk on the development of peptide-based hydrogels for cancer therapeutics. So this is work that he's published in Nature Nanotechnology, and it has been done in mesothelioma. And our second talk was from the wonderful Dr. Greg Lazinski. We all know Dr. Lazinski from Emory, and he gave a talk on adoptive cell therapy for cholangiocarcinoma, focusing on CAR T cells and CCA TIL products. And then we heard, so I'm so grateful to our European colleagues from coming all the way here. And so we heard from Dr. Dieter Sauer, who's at the German Cancer Research Center. He had a wonderful paper a couple of years ago in Cancer Discovery describing a novel model of extrahepatic cholangiocarcinoma. So as many of you know, that many of the majority of the preclinical models of cholangiocarcinoma are intrahepatic models. And so the Perry, Heiler, and Distel have really been neglected when it comes to preclinical models. And so I think Dr. Sauer's contribution was very meaningful in this regard and describing a context-specific model of cholangiocarcinoma. And then our next session was the genetic and epigenetic drivers of cancer, are moderated by Dr. Sia and Dr. Stanger. And we heard the first talk was on epigenetics of cancer in cancer, and using single-cell approaches, single-cell epigenome in cancer. And so this talk was given by Alexander Maya, who's a rising star in epigenetics in cancer biology at Mayo Clinic. His work is primarily focused on breast cancer. And then our next talk was by Dr. Katie Kelly from UCSF on germline genetics and risk of cholangiocarcinoma, where she really provided a very beautiful and comprehensive overview on testing as well as screening approaches when it comes to germline genetics. And then we heard from Dr. Daniela Sia, who had a talk on genetic alterations and need for an integrated approach in the different subtypes of cholangiocarcinoma. And I built on that talk to define the tumor immune microenvironment correlation with the tumor genotypes. And I think what I'm missing here, and I apologize because that was also in our first session, it was a talk by Dr. Rory Smoot, who's also at the Mayo Clinic, on integrated multi-omics, where he has a data set that he has comprehensively analyzed and described molecular subtypes that correlate with survival. And then we had a state-of-the-art lecture from Dr. Tim Gratton at the National Cancer Institute titled From Mice to Men and Back to Mice, Basic and Translational Research in Cholangiocarcinoma. And this was really a tour de force presentation that covered a large number of preclinical studies, correlated that with findings in clinical trials, and then going back to the bench to see how we can delve deeper into what we find in our patients. Our next session, session C, was recent advances in cholangiocarcinoma biology. Moderators were Dr. Greg Gores and Funda Merrick Bernstam. And Dr. Merrick Bernstam gave the first talk, which was on mechanisms of resistance to FGFR inhibitors, which we know that is a really important area of investigation currently. And we heard from another one of our German colleagues, Dr. Michael Dill from the German Cancer Research Center, who gave a talk on a novel FGFR2 fusion-driven model of cholangiocarcinoma, and highlighting the importance of having relevant preclinical models that allow for systematic immune population analysis as well as synergistic drug testing. And then we heard from Liberalis Voila, who is a postdoctoral fellow in Sida Kugel's lab at Fred Hutchinson Cancer Center, and he gave a wonderful talk on SRC inhibition and working via the PP2A complex. So if you remember from this morning, Dr. Voila is also one of our 2024 Research Fellowship awardees. So we really look forward to seeing his career unfold. And then we heard from Pedro Rodriguez from Spain. He works with, and many of you know TUS, he works with TUS Spinalis, and he gave a talk on biomarkers and screening approaches for early detection of cholangiocarcinoma. And our last session, but not in terms of the robust discussion we had at the end of the day, it was a really engaging session on emerging human microphysiologic models. Moderators were Dr. Anna Leo from Italy and Dr. Bolter from the UK. And the first talk in that session was on 3D organoid platform approaches from Dr. Benjamin Hopkins from Cornell. And then we heard from Dr. Zhen, who is at MD Anderson and also has a company, Terasaki, and he gave a talk on microorganosphere or MOS technology and using that technology for high throughput drug testing and using patient tissue. And then Dr. Anna Leo gave a very complimentary talk on cholangiocarcinoma on chip. This is work that she has really made a lot of contributions to in the field, and I believe this is work that's that has been published in JHEP reports. And then our last talk was from Dr. Luke Bolter, who gave an overview of ex vivo versus in vivo approaches for modeling the human disease. So as I said earlier, we're so grateful not only to CCF and leadership and the staff, but also to the scientific community for participating in the session, flying from all over the world to give wonderful talks, moderating the sessions, and really, I think, making it a big success. So we're very grateful for that. So we will move on to the 2023 fellowship presentations.
I just wanted to say a couple of words about the fellowship program. I think everyone in the room would agree that more research is needed to move the needle on this disease. And I think most people would also agree that more funding is required to enable that research. But even either of those things are not enough. In addition to having money, you need to have people on the ground making those advances. This is a rare tumor, as we've heard many times, and a very small research community. So the goal of this fellowship program is to create a pipeline, to really create the next generation of researchers that will create a critical mass, that will be able to collaborate together, that will be able to fuel the discoveries that will enable clinical trials and the next generation. As I mentioned this morning, the foundation has been offering these fellowships since 2015, and I've been involved ever since then. And the quality of the grants and the grantees and the science has just continued to get better and better. So it's really a testament to all of you and to the foundation that there really is now a pipeline, that there really is a community of young researchers that are going to make an impact on this disease, I have no doubt. The fellowships are particularly designed to fund individuals who are well into their postdoctoral fellowships, so don't yet have a faculty position, or those who are very early in their faculty jobs. And you'll hear from Dr. Ilyas, who will introduce last year's awardees, who can tell us what they've been up to.
Thank you, Ben. So our first fellowship presentation will be from Yuanli Zhen, and who was a recipient of the Mark R. Clements Fellowship Award last year. And this, Yuanli is from Massachusetts General Hospital, and the title of his presentation is Mobilization of Anti-Tumor Immunity in FGFR2-driven Cholangiocarcinoma.
Okay, good afternoon, everyone. I'm Yuanli Zhen from Nabil Bardisi Lab at MGH Cancer Center. It's my great pleasure here to present my work, and which is, understanding the FGFR signaling in intrahepatic cholangiocarcinoma. As you know, FGFR2 fusion is, FGFR2 alterations are common in intrahepatic cholangiocarcinoma. And in normal cells, FGFR2 activation requires the binding of FGF ligands. FGFR2 alterations in cancer, including gene mutations, results in constitutive FGF ligand-independent activation of FGFR2 signaling to stimulate the tumor growth. And of note, the rate of fusion in intrahepatic cholangiocarcinoma is greater than any other major tumor types. So in the past 5 years, there have been 3 pan-FGFR inhibitors has been approved by FDA to target advanced cholangiocarcinoma with FGFR alterations. And recently, FGFR2 selective inhibitors has been developed. A great majority of patients have been derived clinical benefits. However, in terms of the resistance and tumor shrinkage, and also the drug toxicity, there are still room to improve the therapeutic response. And one big challenge is the inevitable resistance that occurred due to acquired FGFR2 mutations. Here, one sequential blood sample from one patient indicates that the emergence of multiple such mutations. And according to the structure showed on the right, many different mutations that arisen can disrupt the drug binding ability. In addition, so-called bypass mutations that activate the downstream MAPK signaling, like RAS mutation, has also observed in the same patients, indicating there are multiple resistant mechanisms exist in the patients. Beyond this, there are around 25% of patients that develop resistance with unknown mechanisms. Given the complexity of the resistance, it's important to study the biology of FGFR signaling in ICC to develop treatments that could be combined with FGFR inhibitors. And the work we published in 2022, we took an approach that to identify the strategy to potentiate the FGFR inhibition by performing high-throughput drug screen on multiple patient-derived ICC models. And in particular, we were looking for the drugs that could convert the FGFR inhibition from the stopping the cell growth to inducing cell death. And we found that the inhibitors targeting Y-type EGFR could benefit the FGFR inhibition across all the models we tested. And here we show one example that how the EGFR inhibition could benefit the response to FGFR inhibition. And we see that the combination treatment synergistically induce programmed cell death, which is also called cell apoptosis, and durable inhibition of MAPK and mTOR signaling. Importantly, the combination treatment could induce tumor shrinkage and tumor cell arrest in vivo models. We have tested the combination treatments in a large amount of ICC models, and they all showed very effective, and including those sensitive to or resistant to FGFR inhibitor alone. And recently, the research-based clinical trial is open. And my ongoing work aims to more deeply understand the biology of FGFR2-driven ICC to provide the strongest foundation to develop future therapies. And in particular, I have thought to take an integrative approach, spanning from identification analysis of the FGFR targeted genes to identify the FGFR kinase activity by phosphoproteomics. And informed by those studies, we were led to study the function, the impact of FGFR signaling on cell metabolism as well as tumors' immune interactions. First, our expression studies across multiple models have revealed that transcriptional control of glucose metabolism as a key output of FGFR2 fusion. There was a selective inhibition of FGFR's of glycolytic genes, and also accordingly, we see that the decreased glycolysis based on the metabolomics. Moreover, we saw the dramatically impaired glucose uptake in both in vitro and in vivo. And here I show the FDG PET-CT measurement of the glucose uptake in xenografts. Consistently, we saw the reduced glycolysis by measurement of the extracellular acidification rate. By contrast, the oxygen consumption rate was sustained. indicating there was no change on mitochondrial respiration. So the adaptive metabolic shift could sustain the cell viability response to the oncogene-activated metabolic reprogramming. Here we saw that the FGFR, the impairment of glucose due to the inhibition of FGFR signaling is compensated by the increased fatty acid dependency and also the fatty acid oxidation rate. This was associated with increased liberation of stored fatty acid and by lipolysis, as shown by a decreased number of lipid droplets. Our data also suggested that activation of a nutrient sensor, AMPK, might have promoted the shift, as shown by progressive activation, increased activation of phosphorylation of the key lipids at the AMPK sites. Last, we are asked whether the compensatory mitochondrial changes upon FGFR inhibition could be therapeutically exploited. So here we use an inhibitor of mitochondrial quality control, known as ONC212, combined with FGFR inhibitor. ONC212 can potentiate the FGFR inhibitor efficacy, and the effect could be increased by fasting, which further reduces the glucose availability in the tumor. And here you can see the effect on the tumor volume change and also the tumor cell proliferation. So we think we are building a picture of complex biological change caused by the FGFR2 signaling in ICC. But perhaps the most important area for the development, therapeutic development is identifying the impact of FGFR signaling on tumor immune interaction. And our ongoing work is addressing this problem. So to study the tumor immune interaction at each step of tumor development, we have established a genetically engineered mouse model of ICC by conditionally activable FGFR2 fusion, knock-in allele crossed to a P10 knockout strain. And the Zhen model recapitulates the histopathology feature of ICC patients. Moreover, our preliminary data indicates that the model is sensitive to FGFR inhibition and thus amenable for future studies of the biology of therapeutic response. Our studies for the molecular and cellular basis of FGFR inhibitor response is underway. And the data to date indicate that the FGFR inhibition trigger interesting changes on immune microenvironment with profound CD8 T cell infiltration. And this was linked with upregulation of type 1 interferon response in tumor cells, which we were studying functionally. And so we think this and other encouraging findings have really reinforced the interest in deeply exploring the the impact of FGFR signaling on tumor and immune interaction. And our future work will take multiple technologies to understand the impact of FGFR signaling on tumor immune interplay in the Zhen model and also patient samples to inform potential immunotherapy. With that, I would like to thank my mentor, Nabil Bardisi, and all the lab members. Thanks to the grants to support the research and all the patients and their families who are not just the starting point of the research, but also the ultimate purpose. And in the end, I would like to acknowledge Mark Clement Memorial Research Fellowship to support my research last year. Thanks.
Congratulations, Yuanli. That is really impressive work, and what a tremendous example of the quality and high-impact science that the Research Fellowship Program is supporting. Because we have 8 presentations, we won't have time for questions, but please feel free to, during the break, to find the awardees and interact with them. So our next talk is by Caitlin Conboy from the Mayo Clinic, supported by Susan G. Morrison. The Susan Morrison Research Fellowship was awarded to Caitlin to support her work on epigenetic dysregulation in cholangiocarcinoma.
Thank you so much for the opportunity to present my work. I want to start with the context that there has been tremendous progress in biliary tract cancer with the first approved targeted therapies in the last 4 years for a subset of patients who have particular molecular alterations. But despite this, right now, most of our patients, the majority, still don't have any targeted therapy options. And so I and others in this research community are really motivated to find the molecular mechanisms and therapeutic vulnerabilities of other molecular subtypes, other driver mutations. I'm focusing on the switch SNF subunits. These are mutated overall across anatomic subtypes in about 30% of biliary tract cancer. And the role of these complexes in cancer has really only been appreciated in the last 15 years, and we're just beginning to understand how that functions and how to treat it. It's really beyond biliary tract cancer, with about 20% of human cancers overall having mutations in these subunits. And so if we are able to develop treatments for this, it will have an impact on a lot of patients. This is a diagram of 2 of the switch SNF complexes called BAF and PBAF. These are molecular machines that remodel the chromatin by moving or ejecting nucleosomes. And that has consequences for opening up the chromatin, allowing gene expression, and it affects DNA repair, cell cycle, and other cell processes. I'm highlighting some of the most commonly recurrently mutated subunits, ARID1A, I'm going to try to use a pointer, which is a participant in the BAF complex and mutated in about 18% of cholangiocarcinoma, and PBRM1, which participates in the PBAF complex and is mutated in about 10%. These 2 are the focus of my project. Interestingly, when you look at the genetics of human tumors, these subunits are co-mutated in the same tumors. I'm highlighting the significantly co-mutated pairs in this diagram, and you'll notice that ARID1A and PBRM1 are actually co-mutated in about 4% of biliary tract cancer, which is something I'm interested in. I also wanted to look at what oncogenic drivers co-occur with these mutations, and activating mutations in PI3 kinase, but not other oncogenes, are associated with mutations in ARID1A and PBRM1. And so on the basis of the human tumor genetics, the hypothesis was that ARID1A and PBRM1 might function as tumor suppressor genes that cooperate with each other and with AKT activation to drive cholangiocarcinoma formation. And to address this, we took the approach of generating multiple novel mouse models of cholangiocarcinoma initiated by knockout of ARID1A, PBRM1, or both, along with AKT activation in the cholangiocytes. From that, we derived a new mouse cholangiocarcinoma cell line called APAC2 and a syngeneic orthotopic tumor model. And these are important models because they can be used to understand the biology of the tumors and to test potential treatment approaches in the lab before trying to bring them to patients. This is a schematic diagram showing how these mouse models were made. We're trying to alter these genes in the cholangiocytes, and we use a surgical method for we call biliary installation to transfect the cholangiocytes with plasmids. I'm showing those on the left here. Knockout of ARID1A or PBRM1 was accomplished with plasmids expressing 2 guide RNAs per gene. AKT, which is downstream of PI3 kinase, was activated with myristylated AKT on a transposon, which is integrated into the genome in the presence of Sleeping Beauty transposase. This plasmid cocktail is mixed with lipid, put into the biliary tract, and the mice are monitored for tumor formation. We sacrificed them at 8 months after surgery. The tumors that formed at that time, we analyzed. We were also able to dissociate and clone them to form cell lines for reimplantation back into black 6 mice. Here I'm showing example livers from 3 cohorts of mice. The top here is APAC mice, which have all 3 genes altered, knockdown of ARID1A, PBRM1, and activation of AKT. AAC, which only has ARID1A knockdown with AKT, and PAC, which only has PBRM1 knockdown with AKT. Tumors formed in all 3 subtypes. Oops, sorry, wrong number. Here's the graph of the nodule formation. We have a small number of mice so far, but the trend that we're seeing is that ARID1A appears to be a more potent tumor suppressor gene, with the mice in those cohorts having more tumors. And there appears to at least be a trend towards some cooperation and increased tumor genesis in the combined APAC group, but we're confirming that trend with additional mice. The tumors that form here are cholangiocarcinomas, confirmed by expression of cytokeratin, and they also do have fibroblasts densely infiltrating the tumor, shown with alpha smooth muscle actin staining. And this is similar to the human tumors, which have a dense stroma as well. So these studies have shown that ARID1A and PBRM1 are bona fide tumor suppressor genes in the biliary tract. I'm skipping the cell lines. From this APAC tumor, we actually dissociated it and cloned out a cell line called APAC2, which is cytokeratin positive. Importantly, we did confirm that it has knockout of both ARID1A and PBRM1 compared to 2 control cell lines that you're seeing there. SB1, which is a mouse cholangiocarcinoma cell line, and DIA, which I'm going to talk a little bit more about on the next slide, is a pre-malignant, non-malignant mouse cholangiocyte cell line that only has activation of AKT. When we put the APAC cells back into the liver of a black 6 mouse, they do form tumors which retain cytokeratin expression, and similar to the primary tumors, they recruit in fibroblasts stained by alpha smooth muscle actin. So these are really useful preclinical models that do represent the genetics that we see in the human tumors, and I hope that they'll be helpful tools to test treatments going forward. But then the question is, how are we going to treat them? And so the next question was, what pathways are activated in these ARID1A and PBRM1 deficient cholangiocarcinoma that could potentially be targeted? And we have so far just taken the basic approach of transcriptome analysis with bulk RNA sequencing, comparing the APAC cholangiocarcinoma cells to those DIA cells, which are pre-malignant and only have AKT activation. And this is a heat map showing differential gene expression of about 2,000 genes using these cutoffs. In the APAC cells, there's loss of expression of several tumor suppressor genes like CDKN1A and E-cadherin. And we were interested to see the most highly upregulated gene was periostin, which is a glycoprotein that's abundant in the extracellular matrix of desmoplastic tumors. And it can signal through integrins and RTKs in cancer cells. It's actually known to be upregulated in cholangiocarcinomas, although it's generally thought to be expressed by the fibroblasts rather than the tumor cells, as we're seeing in this case. Sorry, and just to go back and say, this is something that I'm investigating further. We're looking at the periostin expression in some of the human model, in some of the human samples, and doing some knockout experiments to see if it's directly regulated downstream of loss of function of ARID1A and PBRM1, and to see if this model depends on periostin for its growth. That's ongoing work. In the last 1 minute, I'm going to very briefly tell you about a parallel approach we're taking to try to identify unique dependencies of the switch-SNF mutated cholangiocarcinoma. We're taking advantage of publicly available data from Dependency Map to analyze synthetic lethality in human biliary tract cell lines that have mutations in the switch-SNF subunits. If you're not familiar, Dependency Map is a large-scale effort led by the Broad and Sanger Institutes to characterize molecular alterations and dependencies across human cancer. This data they've generated has included genome-scale CRISPR screens, which help us to identify genes that are selectively essential in particular cell or mutational contexts. This included 35 biliary tract cancer cell lines, 14 of which we found have deleterious loss-of-function mutations in switch-SNF subunits. And I'm comparing those 14 cell lines to 17 that were non-mutant for any subunit. There were some others that got thrown out for indeterminate changes. And what I was looking for is genes that are selectively essential in the cancer cell lines that have switch-SNF mutations. So I'm not interested in genes up here that are totally non-essential, which is most genes, or genes way down here with a really strong gene effect that are just lethal in every cell line, regardless of mutation status. I'm looking for the genes that are more lethal in cell lines with these mutations. And this is a volcano plot showing those genes with selective that are selectively essential on the right. These genes are enriched for metabolic pathways, including nucleotide sugar metabolism, and UXS1 is one of the targets that I'm focusing on a little more. I realize I'm now over time, so I will not focus on this other than to say we think UXS1 is important in sugar nucleotide metabolism, and we think it could be a selective dependency, and this is something that I'm following up on. So just to conclude, what I've shown you is novel mouse models of ARID1A and PBRM1 deficient cholangiocarcinoma based on the human tumor genetics that I'm using to try to develop new therapeutic approaches. I'm showing you two candidates that I've focused on so far, but in the next year I'll be working on further validating those targets in other systems and using the models that I've made to do preclinical testing in vitro and in vivo. So I want to thank my cholangiocarcinoma family at Mayo, including my mentor, Dr. Gores, and Jayla Millender, who's down on the lower corner, who's a wonderful technician that I work with. And I really want to thank the Cholangiocarcinoma Foundation, and in specific, the Susan G. Morrison for supporting the fellowship that allowed me to do this research over the last year. And I'm really grateful to the foundation for having another year of funding to continue this project. So thank you.
In memory of Marian U. Schwartz, CCF supports the Marian U. Schwartz Memorial Research Fellowship Grant, and in 2023, this was awarded to Ethan Ludmir from MD Anderson Cancer Center, whose focus is the integration of radiomics and immunogenomics to predict tumor-related liver failure for patients with metastatic intrahepatic cholangiocarcinoma.
Getting oriented up here. Good afternoon, everyone. A pleasure to be here today. My name is Ethan Ludmir. I'm from the Department of GI Radiation Oncology and the Department of Biostatistics at MD Anderson Cancer Center. And it's a privilege to be talking today about expanding horizons in the role of radiotherapy for advanced intrahepatic cholangiocarcinoma, integration of immunogenomics, radiomics, and expanding the frontiers from there. For something completely different compared with the prior terrific two talks, external beam radiotherapy for intrahepatic cholangiocarcinoma is an effective local therapy. It's advantageous in that it's non-invasive, it can treat large tumors, it can treat tumor thrombi, and a series of retrospective and prospective, including real-world data sets, have confirmed that with ongoing technological developments, use of ablative high-dose radiotherapy can improve outcomes for patients with unresectable localized intrahepatic cholangiocarcinoma. We're entering an exciting time in the multidisciplinary space as we learn better how to integrate multimodality therapy for patients who previously were ineligible for this sort of an approach. Rather than receiving serial line of systemic therapy after systemic therapy, we're learning better how to integrate multimodality therapy even in the metastatic setting. Cholangiocarcinoma is a terrific teacher in this regard. Cholangiocarcinoma's proximate cause of death for the vast majority of patients is tumor-related liver failure, primarily driven by biliary and vascular complications from the primary tumor itself. Notably, that proportion of patients, 70 to 80%, remains that level even among patients with extrahepatic metastatic disease, that is, disease that's disseminated from far beyond the liver. This still remains the primary driver and cause of death, which begs the question, if we thoughtfully deliver local therapy even in patients with metastatic disease, could we theoretically improve oncologic outcomes with local control leading to improved overall survival? To that end, at our institution, we have treated patients with extrahepatic metastatic disease with multimodality therapy using ablative dose RT to patients with dominant intrahepatic disease, and demonstrated that for those patients who did receive ablative dose RT, we see marked improvements in overall survival. Now, of course, it's a retrospective series, but even controlling for the itinerant limitations of selection bias through propensity score matching, through landmark analyses to account for immortal time bias with multivariate analyses, and using comparisons with real-world data sets, we see the persistence of a substantial improvement in overall survival for these patients. And as proof of concept, this is driven by reductions in the incidence of tumor-related liver failure. Patients who are treated with systemic therapy alone, even in the modern era, have an 82% rate of death due to liver failure proximate to the primary tumor. However, when you add in local therapy with ablative dose radiation of the liver, we see this number drop to 47%, and indeed the time to tumor-related liver failure for those patients increases from the median time of 9 months to 18. Now, this is all well and good and hypothesis generating, but can we better understand which patients are optimally to be selected for use of multimodality therapy, even in the metastatic setting? To that end, we sought to integrate radiomic features, genomic features, and immunological features as we understand which patients benefit from multimodality therapy and how. To that end, here I'm showing you examples of patients who have high or low vascular enhancement of tumors. They're shown in different phases, but it's to kind of demonstrate the example itself. And what we can see is that by quantifying pre-treatment vascular enhancement, patients who have hyper-enhancing tumors seem to do better than their hypo-enhancing counterparts. And what's more, we see an increased treatment effect size for the addition of radiotherapy for those patients with hypervascular tumors. And there are implications here as well for our colleagues in IR who deliver transarterial therapies. So not only do we see the prognostic role of pre-treatment vascular enhancement, but we also see differential treatment effect, with the addition of local therapy being most impactful for patients who have hyper-enhancing tumors. In terms of how we assess therapeutic response, we're moving beyond RECIST-based measures, which look at anatomic measurements, are in fewer dimensions, and transitioning towards quantitative, volumetric, that is to say three-dimensional, enhancement as a measure of treatment effect. And if we looked at this volumetric Q-EASL, which has been shown and validated in other disease sites in the liver, we can see that those patients who receive effective local therapy have Q-EASL responses. We're seeing that here in the top panel, where we have pre-treatment and post-treatment images of a patient who had response, and you can see the heat map cools off accordingly. And on the other side, a patient who did not respond to local therapy, where you see increased evidence of volumetric enhancement after the therapy was given. Very notably here, as we think in the room here are many clinical trialists and prospective clinical trialists, that RECIST-based measurements offer poor discriminatory value in looking at how patients are going to perform with the addition of both systemic and with local therapies. On the other hand, by using Q-EASL-based volumetric enhancement response, we see marked improvement of discrimination based on patients that will do well and patients that will do poorly, and those are overall survival curves there. As we integrate, and we've heard terrific talks on this subject already, the integration of molecular and genomic profiles, we find that certain mutations might be associated with differential radiomic characteristics and collectively contribute to a specific prognostic or predictive picture. ARID1A mutated cholangiocarcinoma is associated with inferior overall survival, but we also see differential changes in treatment response based on Q-EASL based on ARID1A mutational status. ARID1A mutated cholangiocarcinoma demonstrates poor Q-EASL-based responses compared to those patients with ARID1A wild-type disease. Taking it one step further, now in the era of Topaz and with Keynote, we see that if we look at multiplex immunohistochemistry of the central tumor from biopsy specimens, immune cell infiltration is associated with differential radiomic features. Patients that have poor, low volumetric enhancement are associated with immune cold tumors. These patients are more likely to be immunologically colder, whereas immunologically warmer phenotypes are more associated with patients that are more likely to respond radiomically. And we see this borne out based on recent data published by the group at MD Anderson if you correlate with immune cold subgroups. So as we pull together different omic data, radiomic, immunogenomic, and further data, we see that each of these individually associates with each other, with prognosis, and with treatment response. And as we look to the future, we're building unified models that better understand how we can discriminate patient responsiveness and which patients should be selected for differential receipt of multimodality therapy. We have an ongoing mathematical model that's also furthering development of personalized radiation dosimetry and receipt. But beyond these, we see the expanding role of the multidisciplinary team and the integration of local therapy even in advanced disease. Patients with supermassive tumors, patients with metastatic disease, we are understanding better that metastatic disease itself is not a purely systemic problem, and thoughtful integration of multimodality approaches can meaningfully improve quantity and quality of life for patients. And with support from CCF through this fellowship and others, we're preparing to launch the Hyperion cholangiocarcinoma trial, which is a randomized phase 2 study assessing the role of local liver-directed therapy to the dominant tumor of patients with advanced intrahepatic cholangiocarcinoma. And I think critically for this audience, a patient-centered audience, the trial is patient-centered in its design, with a primary endpoint of overall survival and a key secondary endpoint of hepatobiliary specific related quality of life. And so as we look to the future, it's exceedingly bright as we bring everyone into the room, a multimodality and a multidisciplinary approach as we think about how best to serve cholangiocarcinoma patients. So with that, I'm exceedingly grateful for colleagues and mentors, Eugene Coy, Milin Javli, Yuanli, many of whom are present. I'd also like to express profound gratitude to patients, their families, and of course to the Cholangiocarcinoma Foundation. This work has been made possible by the Marianne U. Schwartz Memorial Research Fellowship, and it's only through support from CCF that we're able to advance this research, open prospective clinical trials, and meaningfully move the needle for patients with cholangiocarcinoma. Thank you.
Thank you, Ethan. In memory of Stuart Mathur, who passed away from the disease in 2020, his wife Colleen and the Mathur Family Foundation are supporting the Stuart Mathur Memorial Research Fellowship Grant. And in 2023, this grant was awarded to Emilie Warren from Emory University, whose focus is CD4 positive, CD26 positive tumor-infiltrating lymphocytes, and how this population represents a novel strategy to improve the efficacy of adoptive T-cell transfer therapy in cholangiocarcinoma.
Thank you so much to the meeting organizers for the opportunity to present my work today. I'll be discussing a series of novel techniques to improve adoptive cell therapy products for cholangiocarcinoma today. These are our disclosures. Adoptive cell therapy, or ACT, using tumor-infiltrating lymphocytes, or TIL, is a cellular-based immunotherapy in which tumor tissue from the patient is removed, and the cells present in that tumor, which include any immune cells, are then put into a culturing system in which the T cells preferentially grow. The patient then will ultimately receive back a concentrated infusion of these cells. And in recent work, where patients with advanced melanoma were treated with TIL versus checkpoint inhibitor ipilimumab, there was a significant improvement in progression-free survival in those patients who received the TIL. And what you can see in those waterfall plots on the far right, where each bar represents a patient, and a patient who received or who actually responded to the therapy being represented by a bar that dips below the x-axis, you can see that in the top half of that graph, where patients received TIL therapy, there were many more patients who responded than to those who got checkpoint therapy. Based on these very exciting results and others, a couple of months ago, the FDA approved the use of TIL for advanced melanoma. In cholangiocarcinoma, however, this therapeutic option really has yet to be widely studied. Although the remarkable case of a patient who was treated with TIL as part of a clinical trial really shows us and gives us hope that there's promise in this disease for this therapy. Genomic sequencing that was performed on her tissue allowed for the identification of a subset of T cells that were specific to unique proteins expressed by the tumor. And as you can see in the graph on the far right, when this patient was infused with a solution that contained those mutant-specific T cells, the patient ultimately achieved a durable remission of disease. And while these results are striking, much more work remains to be done to optimize this therapy for cholangiocarcinoma. And so in the preclinical setting, our lab has worked to develop a mouse model to further study cholangiocarcinoma TIL. For these experiments, immunocompetent black 6 mice were inoculated with a mouse-derived cholangiocarcinoma cell line called URCCA 4.3. These tumor cells were either injected subcutaneously or directly into the liver. And after a period of up to 3 weeks, these tumors were excised from the animals and the tissue digested into a suspension that contained both tumor cells, but also the immune cells that were present. And within that suspension, then we could analyze the properties of each individual cell using fluorescently tagged antibodies. So what you see on these graphs represents data, each single dot is representing the data of a single cell in which antibody tags then are present. And from both the subcutaneous and liver tumors, although we know that these are tumors that traditionally have low T cell infiltration, we did find that there were both CD8 and CD4 T cells, but there was more CD8 than CD4, about a 15 to 20% difference versus about 10% of the total immune cell population. And one of the very talented MD PhD students who works in our lab, Megan Whitling, has done a more comprehensive analysis into all of these different immune cell subsets in the liver tumors, and she's presenting a poster at this meeting. Once the tissue from the mouse has been digested, we then perform a selection for cells that are CD45 positive, and this is a universal marker for immune cells, in order to exclude any of the tumor cells going forward. These CD45 cells then are put into a culture dish, where traditionally they are expanded in the presence of activating growth factors CD3 and cytokine IL-2. And it is really this phase of the process that I'm going to talk about a lot today, where we have a huge and tremendous opportunity for our lab to test various strategies with the aim of making a superior TIL product. One such strategy is to select for a population of T cells with favorable anti-tumor properties. Our lab has previously identified such a population, termed type 17 cells. These secrete IL-17. They have stem cell properties. They're known to express high levels of CD26, which we've also shown to mark cells that have enhanced anti-tumor efficacy. And in mice with melanoma tumors, we've previously shown that adoptive cell therapy with these type 17 cells significantly reduced tumor growth compared to animals that were given T cells expanded by traditional methods. Francie Bennett is another research fellow working with me in the lab who gave a talk about this earlier today, and she also has a poster presenting this week regarding further information about how we expand these type 17 cells. We hypothesized then that these type 17 cells should show anti-tumor activity in mice with cholangiocarcinoma tumors. So we generated the type 17 cells as I previously described, where we use a specially designed cocktail in the culture mix in order to enhance or expand these type 17 cells. Liver tumors were then established in a separate cohort of RAG knockout mice. These are genetically manipulated to have no naturally occurring B or T cells. And this time we had cholangiocarcinoma cell lines that expressed luciferase, meaning these cells were luminescent. And this bioluminescence, we could measure externally to monitor growth over time. So the day prior to adoptive cell therapy, the mice were depleted of any circulating immune cells. This is very similar to what we would do in a human patient. And then 2.5 million T cells were infused into the mice, and we were able to monitor tumor burden over time using the bioluminescence measurements. Initially, these animals who received adoptive cell therapy experienced a control of tumor growth, which you see in the red line on the left, compared to those animals who did not receive any therapy. However, by the end of the experiment, these tumors had rebounded to be the same size as controls. And on the right, you see some example images of what the mouse bioluminescence or tumor burden looks like. and what we can quantitate. And highlighted on the far right is an example of one particular mouse who appeared to have initial tumor regression followed by progression at the end of experiment. The promising finding was that when we did examine the tissues of these mice at the very end of our experiment, we saw that the T cells we infused into the mouse were actually still present after several weeks. As you can see on the far right curve, the control mice, these don't have any T cells and didn't receive any therapy. So in the treated mice, any detections that we see truly are the T cells that we initially gave. However, the greatest number of T cells ultimately were found to be stuck in the tumor-draining lymph nodes, and a very low percentage was found to have infiltrated into the tumor. Interestingly, although we started, if you recall, with a predominantly CD8 positive T cell population, the majority of what remained behind was CD4 positive. Certainly there are likely a multitude of reasons that these type 17 cells fail to infiltrate and kill a cholangiocarcinoma tumor. And one hypothesis that we had was a lack of tumor antigen specificity. So Megan devised a novel system that would include some dead tumor cells, and we had additional antigen-presenting cells that we introduced into the culture mix from mouse spleens. The presence of these dead tumor cells would allow for their antigens to be processed, those specific tumor antigens to be processed and presented to the T cells by the antigen-presenting cells, and that these specific T cells then would be the ones that would preferentially expand. This time before we went into the mouse, we used a cell impedance-based cytotoxicity assay to assess tumor killing. And the tumor cells were plated onto dishes that had electrodes embedded into the bottom of the plates. So when tumor cells adhere to the bottom of the plate, and when those are killed off by the T cells that we treat them with, the impedance change across the electrodes is a detectable difference that we can quantitate. When we take a closer look first at the characteristics of these cells, we saw that the CD4 T cells that we expanded actually had increased expression of markers associated with the activation of antigen-specific T cells. And there was also decreased expression of the checkpoint molecule LAG3 in both of the CD4s and CD8s, but their cytotoxic functionality largely remained unchanged. So this experiment, we had gone back to the traditional culture of just expanding these TILs in the presence of IL-2. And what you're going to see are graphs showing what we quantitate from our cell impedance-based cytotoxicity assay, which is % of tumor cells that are killed by the tumor, by the T cells. So when there was a 1-to-1 or 5-to-1 ratio of T cells to the target tumor cell, the tumor killing remained very low up until once we added 10 times as many T cells to tumor cells, then we finally saw some impressive tumor killing. But when we took our new method of expanding T cells in the presence of tumor antigen, there was nearly 100% tumor killing achieved, even at this very low 1-to-1 ratio. What I've shown today is really just a promising beginning, and we're increasingly finding that likely these CD4 positive T cells are an important subset that we need to address further. Specifically, our lab has particular interest in looking more closely at these CD26 expressing CD4 positive T cells. Additional strategies include the use of CAL-101, which is a PI3 kinase inhibitor drug that we've previously shown can decrease exhaustion and improve mitochondrial function in T cells when they're expanded in the presence of this drug, as well as improving T cell infiltration into tumor by genetically engineering them to express certain chemokine factors. Certainly, the ideal TIL product for cholangiocarcinoma will likely be generated from some combination of these strategies. So I'd like to acknowledge my clinical research mentor, Dr. Shishir Maithel, as well as both the Lisinski and Paulos labs for their contribution to this work, particularly to both Megan Whitling and Francie Bennett, who truly have been invaluable to this project. We've spent many a long and fun day in the lab together. But above all, I'd like to express my deepest appreciation to Colleen Mather and the Mather family for supporting this research through the CCF Fellowship Program. Thank you.
Thank you very much, Emilie. It's really a wonderful cohort of young investigators at Emory. So in memory of Jacques Dupont, Barbara Dupont family and friends are supporting the Jacques Dupont Memorial Research Fellowship Grant. And in 2023, this grant was awarded to Jonathan Zia from Northwestern University, and the focus of his grant was the integration of multi-omics in Bile for biomarker discovery in primary sclerosing cholangitis and cholangiocarcinoma.
All right, thank you for giving the opportunity to present my research here. Where's the clicker? Next. Oh, next screen. Okay. So yeah, I mean, the diagnosis of biliary malignancy still remain a diagnostic challenge in most clinical settings still. You know, brush cytology is currently the gold standard, and the published sensitivity is 35%, which is, you know, these are studies done at high volume tertiary referral centers with, you know, world experts at ERCP. I think in the community, the realistic sensitivity is probably down to 25%, you know, and if we do things such as increased brushings, FISH, maybe cholangioscopy, we can increase that, but that's not available everywhere. So accurate and scalable identification of malignant strictures is still an ongoing clinical issue. And my interest is really to study this in the form of bile in patients with coming in for evaluation of indeterminate biliary stricture. And one, we want to study that in the microbial signatures because we know the role of microbiome and its role in the development of certain cancers. And the other thing is metabolomics. That's based on the idea that cancer uses different types of metabolites or nutrients compared to normal cells. So as you can imagine, a highly metabolic tumor is likely to have different type of byproducts that can be excreted into bile compared to something that's in benign biliary disease. So for us, we had a prospective, single-center prospective cohort study where we pretty much enrolled everyone undergoing ERCP for, you know, indications such as biliary obstruction, jaundice, abnormal liver enzymes, or evaluation of dominant strictures in PSC, and abnormal imaging. And here we pretty much collected every patient that came through. We collected 3 to 5 cc's of biliary aspirate, either through the sphincterotome or the balloon. And afterwards, we analyzed microbial and metabolomic markers. So for the demographics of this cohort, we had about 140 patients so far. 45% of them are female, 76% are native papillas, median age about 60. For malignant strictures, we included patients with cholangiocarcinoma, pancreatic cancer, and malignant strictures due to metastatic disease. And for benign biliary disease, we had patients with normal cholangiograms, colloidal lithiasis, and transplant anastomotic strictures. And we also started collecting patients with indeterminate biliary strictures as well. So the first thing we did after collection is we looked at profiling the bile microbiome in patients with benign and malignant biliary disease. The first thing we did is look at the taxonomic breakdown. The majority of the included E. coli and Streptococcus. And interestingly, you know, when we looked at the hierarchical clustering of the phylogenetic distances, benign disease tend to cluster together, PSC and the malignant disease cluster together. And one of the first things we looked at is alpha diversity, which essentially is how diverse are the different populations. And we did not actually see a big difference between cholangiocarcinoma and benign biliary disease or PSC initially. But when we looked at the composition, which is looked at by the statistic beta diversity, we saw a pretty big difference between cholangiocarcinoma compared to benign biliary disease and PSC. And next, we looked at, are there any different microbes that are associated with cholangiocarcinoma compared to benign biliary disease or PSC, and we saw there were actually some increases in different bacterial species in cholangiocarcinoma compared to these groups. And functionally, the microbes in cholangiocarcinoma appear to have different types of utilization of nutrients compared to benign biliary disease. So this data initially really gave us a good idea that there might be some type of metabolic differences in the bile of patients with cholangiocarcinoma, and really led to our second point where we wanted to look at different metabolites that are in bile in patients with cholangiocarcinoma. So for this second part, we pretty much, for all the bile we collected, we extracted mainly for initially polar metabolites and then performed mass spec, and we correlated this with clinical data. The first thing we did is we looked at this in a PCA where we looked at different groups in patients with normal cholangiograms, gallstone disease, and cholangiocarcinoma. And interestingly, some of the patients that had cholangiocarcinoma that we collected bile on were people undergoing treatment, and they did kind of group together with the patients that had either benign disease or normal cholangiograms. And next, we looked at, are there any focal enrichment of different types of metabolites that are in the bile of patients with cholangiocarcinoma. And we found that there are 4 pretty unique ones that were both very statistically significant and highly upregulated. And we see here these 2 of them are methylated nucleotides, and 2 are more metabolic markers of urea and butyric acid. They're statistically significant and highly upregulated in the bile of patients with cholangiocarcinoma. And once we increased the study to a larger cohort, we had, I think, over 50 benign biliary disease controls that these elevations were still persistent in the population. So, what's the goal of all this? I think for this, really, can we identify a liquid biopsy, a non-cellular-based approach to risk stratify patients with coming in for an evaluation for an indeterminate stricture? Normally what we do is we pretty much just do ERCP until we brush, until we pretty much get some type of tissue diagnosis. Either they reveal themselves by having some type of metastatic disease or we can biopsy, or the brushing eventually become positive, but that could take a very long time. And I think previous studies, I think based on Cholangiocarcinoma Society, have shown that some people have done epidemiology studies that showed that it could take up to 6 to 10 months to get a good diagnosis, which is pretty long to wait to before therapy, right? So for us, we want to see, can we find any metabolic signatures that can help us risk stratify patients to see who we can focus on. So we have, for here, we had some newer patients where we found 7 indeterminate biliary strictures that we grouped together, and we used the 4 metabolite panel we found, and can we see how accurate they are using patient data we correlated with that we have. So the first one is someone that's 82-year-old. That's the patient on the right there that had an indeterminate stricture. They were referred to me for 82-year-old guy, came in with a TBLA15. We did a lot of ERCPs from 1022 until 1023. We did spyglass, people, he had IR biopsies, everything. None of them were positive until about a year later in November 2023, where finally one of the cholangioscopy biopsies was positive. And then he had a resection, and it was already stage 3 at that point. And you can see here at this point, the sample that was collected in May of 2023, all these markers were already, seems to suggest that they are likely due to cholangiocarcinoma. And another patient here, we had a 68-year-old patient. She presented here initially with a TBLA of 13, had initial ERCP in June of 2023, where we collected bile in that one. And she was found to have a common hepatic duct stricture, where we performed 3 subsequent ERCPs with biopsies. None of them were positive. And finally, her CA19-9 was pre-elevated, and she finally had imaging that showed a peritoneal carcinomatosis, a nodule, where IRA was able to biopsy, and that was positive for adenocarcinoma. But initially, at that initial June collection date, we saw that the bile metabolites for all these markers were seems to suggest that she already has cholangiocarcinoma at that point. And this one, I think, is a really interesting one. This is a 76-year-old patient that had no other medical issue except for she came as a referral for an incidental left intrahepatic ductal dilation. And she had an ERCP with a left main hepatic duct stricture, where we performed 3 ERCPs with cholangioscopy, brush, fishing, everything. They were all negative. I sent her to a surgeon, mainly just because she had pretty good functional status. She didn't have any other medical issues. He saw her, saw the area was very focal, and said, hey, maybe we can just resect this. And he resected it at that point, and it was stage 1 cholangiocarcinoma at that point. And we can see from this data already that, these metabolite panel seems to point that she already had cancer at that point as well. And I think this is a good example of what this type of study could help with. An additional one, I included one of the patients here that was benign, and this is someone that's 70-year-old that was referred with a hilar stricture that was very, very concerning for cholangiocarcinoma. We did 3 RCPs with wire-guided biopsy, with spyglass, fish, everything. They were all benign, and then we checked a CA19-9 that was actually pretty low. And then finally at that point, we checked IgG4 that was elevated. These strictures all went away after prednisone, and it's interesting in this case where all the metabolites were negative and show that they actually did not, have any type of concern for cholangiocarcinoma at that point. So out of these, indeterminate strictures, 3 of them were confirmed benign, and the biomarkers seemed pretty accurate at this point, and the ones that were confirmed to be malignant based on clinical data, the metabolite panel seems to point towards that they were indeed malignant. Overall, I think the bile from patients with cholangiocarcinoma contained distinct microbial fingerprints. and metabolic fingerprints that are distinct, and we can use this to potentially risk stratify patients going forward. So all this work not be done without the help of the Northwestern Interventional GI Lab, and my funding from the Cholangiocarcinoma Foundation, of course, and the ASG Research Award. And thank you.
For our next talk, this is a grant that is in memory of Andrea Scott. Jason Scott family and friends are supporting the Andrea Lynn Scott Memorial Research Fellowship Grant. And I think it goes without saying that without the support of patients and their families, many of these fellowship grants wouldn't be possible. So in 2016, I was a recipient of the Andrea Lynn Scott Memorial Fellowship, and that's a testament to the long-standing support of these wonderful families. And in 2023, this grant was awarded to Jennifer Tomlinson from the Mayo Clinic, whose focus is the role of YAP in cholangiocarcinoma tumor immunity.
Thank you again for the opportunity to present an update on this work. Cholangiocarcinoma is a heterogeneous disease of the biliary epithelium with a diverse molecular landscape and prominent desmoplastic stroma. Due to a lack of symptoms in early disease, patients often present at an advanced stage and thus underlines the need for an effective systemic therapy. Despite the recent discovery and approval of novel targeted therapies for cholangiocarcinoma, response rates remain underwhelming and are only available to a minority of patients. Furthermore, cholangiocarcinoma has high rates of both primary and acquired resistance to systemic therapy, thus underlining the need for a better understanding of cholangiocarcinoma biology. YAP is a transcriptional cofactor that interacts with TEAD and other transcription factors to control the expression of target genes. The YAP-dependent transcriptional program controls organ size and tissue homeostasis by promoting cell proliferation and survival. In normal physiology, YAP is found mainly in the cytosol of mature tissues and is regulated by the Hippo signaling pathway, a series of serine-threonine kinases that inactivate YAP via serine phosphorylation. Cancer cells, however, co-opt YAP programming, switching YAP to its activated form in order to drive tumorigenesis and evade apoptosis. This includes cholangiocarcinoma, where YAP is found in its nuclear and activated form in the vast majority of tumors. However, the molecular mechanism underlying YAP activation is not clear in the majority of human cancers. And furthermore, YAP has been very difficult to target pharmacologically, and this has led to a real shift in focus to the evaluation of other therapeutics in the YAP pathway and the development of novel strategies for YAP targeting. A few examples are included here. So in the first panel, LCK, a Circ tyrosine kinase, was identified as an upstream activator of YAP in cholangiocarcinoma via tyrosine phosphorylation. A small molecule inhibitor of LCK resulted in decreased YAP activity and was therapeutic in preclinical models of cholangiocarcinoma. In the middle panel, therapeutic milk-derived nanovesicles were loaded with siRNA to YAP, its paralog TAZ, and LCK. CCA tumor-bearing mice were treated with these siRNA nanovesicles and resulted in significantly reduced cholangiocarcinoma tumor burden. Finally, in the last panel, those same tumors were taken from mice after treatment with siRNA loaded nanovesicles to YAP, TAZ, and LCK, and underwent immune profiling by flow cytometry. And YAP targeting was found to induce anti-tumor immunomodulatory effects in the cholangiocarcinoma tumor immune microenvironment. So in the exploration of alternative pathways for YAP targeting, we questioned whether TEAD inhibition would be a viable therapeutic target in cholangiocarcinoma. TEADs are a family of transcription factors that are considered to be key mediators of the carcinogenic potential of YAP. In order to perform this work, we utilized a novel small molecule inhibitor named CTX-1009685, or CTX for short, which is a TEAD134 inhibitor with nanomolar affinity by HRTF assay. In order to approach this question, we tested CTX in 2 distinct syngeneic murine models of cholangiocarcinoma, along with multiple patient-derived xenograft models of cholangiocarcinoma, and then finally profiled the immune microenvironment of these tumors after CTX treatment. C57 black 6 mice underwent orthotopic implantation with one of 2 distinct murine cholangiocarcinoma cell lines. Tumors developed over a period of 2 to 3 weeks, and then mice were subsequently treated daily with CTX for 14 days. Tumors in the CTX group were significantly smaller in both models compared to vehicle-treated mice. And qT-PCR analysis of tumors demonstrated decreased expression of YAP target genes. We then moved to testing CTX in 3 patient-derived xenograft models of cholangiocarcinoma, with tumors implanted into the flanks of NOD-SCID mice. In this immunodeficient model, CTX was no better than vehicle in controlling tumor growth. Given the differences between the in vivo models and the previous findings of YAP-driven immunomodulation changes, we questioned whether TEAD inhibition induced changes in the tumor immune microenvironment, and that led to the efficacy that we observed in our syngeneic models. We subsequently profiled the tumor immune microenvironment of cholangiocarcinoma tumors from the previous syngeneic mice treated with CTX. However, minimal signal was detected from this line of exploration. So, as a part of this process, we have so far observed activity in the TEAD inhibitor in syngeneic models, but not in our PDX models, which we thought was initially related to immunomodulation. But to better understand these differences, we're proposing several additional studies which should help us dissect YAP from TEAD signaling in order to identify novel targeting approaches. Some of these future directions include investigation of alternative TEAD inhibitors, including pan-TEAD inhibitor strategies, or the use of siRNA to pan-TEAD in the nanovesicles that I previously discussed. Using the evaluation of TEAD inhibition in the broader tumor microenvironment, particularly looking at spatial transcriptomics and potentially cancer-associated fibroblasts. Looking at combinatorial therapy with TEAD inhibition, including cytotoxic chemotherapy, gemcitabine, cisplatin, or also the use of genetic knockouts of YAP versus TEAD for in vivo studies, including immune profiling. First and foremost, I would like to thank the Andrea Lynn Scott Memorial Research Fellowship for the support in this work. And I'd also like to thank the Mayo Clinic Cholangiocarcinoma Supergroup for their support.
All right, moving on. In memory of Bruce Kopach, who passed away from the disease in December, the CCF board and staff are supporting the Bruce Kopach Memorial Research Fellowship Grant to recognize and honor Bruce's years of service. And in 2023, this grant was awarded to Tin Yun Tang from MD Anderson Cancer Center, whose focus is MTAP loss clonality and targeting in cholangiocarcinoma.
Good afternoon, everybody. My name is Tin Yun Tang. I am a current medical oncology fellow and incoming junior faculty into the Department of Investigational Cancer Therapeutics at MD Anderson Cancer Center. It's really my pleasure this afternoon to present some preliminary findings from my research work investigating MTAP loss in biliary tract cancers. So hopping right at it, what is MTAP loss and why do we care what it is in biliary tract cancers? So MTAP, or methylthioadenosine phosphorylase, is a gene that rather uniquely undergoes a true homozygous deletion. So both of the copies of the gene are lost, and one larger chromosomal microdeletion with neighboring genes CDKN2A, CDKN2B, and on occasion also an interferon type 1 cluster. So these three genes all live on chromosome 9 in the short arm of P in the loci 213. And what really happens is that you have one large deletion of MTAP, CDKN2A, and as well as CDKN2B in one larger event. And very likely the evolutionary advantage of losing the well-known tumor suppressors CDKN2A as well as CDKN2B ultimately leads to a deletion on the alternative allele 2. So in different literature, and mainly genomics literature, you'll hear this referred to as chromosome 9p21 loss. In more clinical literature, you'll probably hear more of it as MTAP loss. But what really happens is that you have a true homozygous deletion, which it has previously been demonstrated in pan-cancer studies to be one of the most common homozygous deletions in a pan-cancer standpoint. In intrahepatic cholangiocarcinoma, we published some data earlier this year really demonstrating that in intrahepatic cholangiocarcinoma specifically, you see this genomic alteration in around 15% of tumors. This really puts this in the ballpark range of other very well and high prevalence mutations in cholangiocarcinoma, such as FGFR fusions, as well as IDH1 and IDH2. You also see this in extrahepatic cholangiocarcinomas, and you also see this in gallbladder cancers. We just don't really have that large-scale data to really derive a incidence estimate for those. And why do we care about MTAP loss in biliary tract cancers? The reason that we care is that more recent TCGA-based studies have demonstrated that 9p21 lost tumors on a pan-cancer standpoint are immunologically cold, generally speaking, and that they have a poor response to immunotherapy. This is, of course, particularly relevant in cholangiocarcinoma, given that some of our more recent advancements in the treatment has been the introduction of immunotherapy into the treatment paradigm. So regardless of whether it is CDKN2A or MTAP that's really driving the formation of this homozygous deletion, what we do know and we have observed is that the fundamental homozygous loss of MTAP really leads to a buildup of its metabolite, MTA. And the buildup of MTA ultimately inhibits a different gene called PRMT5. PRMT5 plays very many different roles in cellular biology, but what has been observed in previous preclinical studies is really that dysregulated PRMT5 leads to dysregulated RNA metabolism, altered RNA splicing, and there have been some distant connections with DNA damage repair as well as genomic stability. Importantly, second-generation PRMT5 inhibitors are already in phase 1 clinical trials, and there's early efficacy signals seen. On the bottom left is a figure published from a paper that came out from Maradi's group that really showed a partial response in gallbladder cases treated with PRMT5 inhibitors as monotherapy. And so my research aim was really investigating specifically this MTAP loss genotype, phenotype, what have you, in cholangiocarcinoma, and then also further understanding RNA metabolism in biliary tract cancer models. So in order to really accomplish this study, I really needed large access to data. And I'm fortunate to be in an institution that has an institutional initiative that is doing very deep profiling of rare tumors and rare cancers, cholangiocarcinoma included. And so what was very nice was that we had a data set on 87 high-quality transcriptomic analysis with matched patient clinical information. There's around 80 patients in this data set, and there's actually very little overlap with previously published data sets coming out of our cholangiocarcinoma research group at MD Anderson. So a lot of this is kind of a newer data set and newer analysis. And so you can really see in this large data set that it's predominantly Caucasian, predominantly intrahepatic cholangiocarcinoma. You have some small incidences of extrahepatic and hilar cholangiocarcinoma, and that very intriguingly, the majority of these samples were taken from the time of surgery. So these were patients that presented with early stage cholangiocarcinomas or slightly advanced cholangiocarcinomas that underwent neoadjuvant therapy and ultimately did arrive at resection. And what the figure on the left is really just meant to demonstrate is that transcriptomic data is extremely high dimension. You have thousands of genes, you have a large amount of patients, and ultimately data is not biological insight. You really need to do additional analysis to really pull anything from this sort of large data set. So when you do a differential analysis, and this was a differential analysis I did based on the MTAP loss patients compared to the intact patients. One of the first things that I really noticed was that actually the MTAP loss patients don't necessarily cluster as much as I would have really thought. But on deeper thinking, this is not necessarily that contradictory to some of our own prior work. I mentioned a paper demonstrating the 15% incidence of intrahepatic cholangiocarcinoma. In that manuscript, what we really saw was that MTAP loss can occur concurrently with other known drivers in cholangiocarcinoma, including FGFR fusions and IDH1 and 2 mutations. So probably the overall genetic signature of MTAP loss may not be so great that you're going to see it overtly on a kind of broad study such as a clustering dendrogram. When you look deeper into the differential analysis, what is also striking about MTAP loss tumors is that you have a downregulation of a great amount of genes, much more so than an upregulation. It's a very, very skewed volcano plot, quite frankly. And when you take a closer look at which of the genes are differentially expressed, actually some of the most strongly differentially expressed ones are long non-coding RNAs, which is kind of interesting when you start thinking about that intersection with RNA metabolism and alternative splicing. So of course, differential expression analysis still too high dimension, still cannot really pull too much biological insight out of this. And so once you do a gene set enrichment analysis, which takes as input a ranked list of genes that you get from a differential expression analysis, you can see on the left really just a snapshot of some of the statistically significant findings. The top 6, the second one is actually a PPAR pathway alteration. So that in more recent time has been associated and linked with dysregulated DNA damage repair. And you can just really see for that walk plot that this PPAR signaling pathway is pretty strongly altered in MTAP loss tumors. So moving on to a different set, though, at this point in time, is that ultimately it's very nice to have large observational data sets, but you're going to need some sort of model that you can investigate and prod and experiment upon. So very fortunately, Dr. Funda Merrick-Bernstam's lab at MD Anderson Cancer Center has pretty systematically been producing PDX models of interesting cholangiocarcinoma patients. And, actually this data set of around 28 PDX models were very heavily enriched in MTAP loss patients. And really what the diagrams here are really just showing is that in intrahepatic and extrahepatic as well as gallbladder, when you see MTAP loss, you get essentially near zero RNA expression levels of CDKN2A, MTAP, as well as CDKN2B, really just validating that if you see a loss on the DNA level, as it makes sense, you also see this loss on RNA level. This has also been correlated with IHC data, which is a little bit more complicated too, because there is some mild disjunction between what you may see on the RNA level and the protein level. But regardless, I'm just really using this data set to introduce the analysis of alternative splicing. So when you think about alternative splicing, there's 3 major analytical forms that you can really do. You can do something called differential exon usage. You can do something called differential transcript usage. What I used in my analysis so far has event-based alternative splicing. And what that really is a little bit of an abstraction of what alternative splicing is. What you do is you essentially count. As in very many high-level omic level studies, you're counting the amount of reads you see in different junctions in different exons. And these are very complicated statistical packages, but what you really see is that you can measure the incidence of 5 different alteration events: skipped exon, alternative 5' and 3', mutually exclusive exons, as well as retained introns. Now, I see, unfortunately, that my figure on the left did not render correctly in this version, but the light green version, the light green over here, right here, represents skipped exon usage. So in both study sets, so on the PDX study set, when I looked at only the intrahepatic cholangiocarcinoma patients, you really see that MTAP loss. You get a very marked enrichment, proportionally speaking, of skipped exon events. Now, of course, this is representing thousands and thousands and thousands of different alternative splicing calls, and that is still too high dimension data to really make sense of. But as an example of what you really see as a skipped exon, very straightforward, you see a skipped exon. And this was, FANCA, of course, is a well-known gene that participates in different DNA damage repair pathways. That was statistically significant to be enriched in skipped exon events. But finally, what you can really do, and the advantage of these kinds of analysis, is that you can further dimension reduce into an overrepresentation analysis. This is a little bit different from a gene set enrichment analysis. This is actually a little bit of an older methodology where you're really looking at what you really see is that you see an increase of splicing events or an alternative splicing events in MTAP loss samples versus wild type in different DNA damage repair pathways. So what you can appreciate is that some of the top hits for gene ontology analysis, which is a little bit different from gene sets. Gene ontology are more general pathways that are not always regulated in the same direction. But when you look at a gene ontology perspective, what you really see is that some of the DNA damage repair are some of the top hits that you're seeing. And I apologize for this figure that did not render correctly in the final version. And the findings so far, just kind of summarizing it all together, is really just that multiple bioinformatic analysis at this point in time really support an intersection between MTAP loss and DNA damage repair. And this potentially provides a rationale for combination targeted therapy and empirical validation with in vitro and in vivo experiments. And it also supports the idea that already in MTAP loss cholangiocarcinoma, these are samples that are already dysregulated from a splicing standpoint. So some of the ongoing work and future directions is really some of the aforementioned in vitro studies with PRMT5 inhibitors and MTAP-less cell lines. And that work is ongoing, and we have some exciting preliminary findings, just not mature enough to share at this point in time. And the additional thing to really do is differential transcript usage analysis with long-read sequencing. The reason why a lot of alternative-based splicing studies at this point in time rely on event-based splicing is it has to do with a fundamental limitation of the short-read nature of the current generation of the next-generation sequencing. To really do an in-depth differential transcript analysis, you're going to need long-read sequencing. And finally, of course, rational combinations of target therapies with PRMT5 really setting up for future clinical trials as the monotherapy arms of these second-generation PRMT5 inhibitors start to mature. So I want to acknowledge all of the people I work with, all the people that give me access to data, all the people that give me insight and direction. It's a long list of people at Emilie that are working on cholangiocarcinoma at this point in time. And I, of course, really want to thank my funder. I really want to thank the Cholangiocarcinoma Foundation. I really want to thank the Bruce Kopic Fellowship, which I was very fortunate to receive.
All right, last but not least, we have the Kathleen and Paul Sindenblad Research Fellowship Grant supported by Kathleen and Paul Sindenblad. And this was awarded to Benjamin Green from the University of Hawaii Cancer Center, and the focus of his grant was peptide vaccine targeting WT1 for immunotherapy against intrahepatic cholangiocarcinoma.
Thank you. It's an absolute honor to be here and present the work that I've done over the last year. So I wanted to basically start out by explaining a little bit about what I do as a cancer immunologist. If you zoom in on cholangiocarcinoma, what you see is a tumor surrounded by normal liver cells. But in immunology, that's not enough, and as we've heard extensively over the last 2 days, immunotherapy is coming a long way and it plays an important role. So really the model that I look at is a tumor that's infiltrated by this rainbow of different immune cells. And if you were to actually zoom in even further and look at an individual cell, what you're going to see is an immune cell, like a T cell, interacting with a tumor cell. And what it's doing is recognizing that tumor cell. It does that in a very specific way. The T cell receptor on the bottom is fitting perfectly into one of the proteins on the tumor cell surface, and it only recognizes the tumor cells as opposed to normal tissue because the tumor cell protein is loaded with something called a peptide. A peptide is simply a fragment of tumor protein. And if that were to happen, the T cell will undergo a process and will destroy that tumor cell. We want this to happen. This is the whole basis of how the immune system can keep a tumor at bay. In immunotherapy, the reason why this is working so well is because we have antibodies that enhance this process from happening. However, one of the problems and one of the reasons why immunotherapy currently isn't working very well is because many cholangiocarcinomas don't have a lot of T cells in them in the first place. And one way to address this problem is with a vaccine. A vaccine, we've been doing this for decades now, trains the body's immune system naturally to recognize either a tumor or a virus and destroy whatever it is vaccinating against. So what we've done is we've taken one of these peptides that are at that interface between a T cell and a tumor cell, and we are trying to develop ways to treat patients with these exact peptides to enhance the amount of tumor immune recognition happening. We've actually done this successfully already. This came out last year in a different type of liver cancer called hepatocellular carcinoma using a tumor protein or tumor peptide called AFP. Unfortunately, this isn't really present in cholangiocarcinoma, but it is in hepatocellular carcinoma. And you can see after we expose these mice to our vaccine, which is based on the AFP protein, and then give these mice hepatocellular carcinoma. On the bottom, you can see what happens with the peptide vaccine. The tumor barely grows at all. Where it does grow, an army of T cells are there present to destroy any cancer cells that might be trying to grow, and these tumors are kept largely at bay. You can see what happens in mice without the tumor vaccine in the control group. Those tumors grow everywhere, and these mice succumb to their disease very quickly. So the question of this project was, can we create a peptide vaccine similarly as we did in hepatocellular carcinoma, but this time in cholangiocarcinoma? Like I said earlier, the real challenge of this work is to choose a different peptide vaccine and choosing our target, because as we learned, AFP really does not work since cholangiocarcinoma cells don't really express AFP. So what we did was we went to the literature and had a lot of debate about which target we were going to select. We ended up going with a protein called WT1, and I know there's been a lot of names of genes and proteins said, so I'm not expecting anyone to memorize this since there's just been a lot of information. But WT1, just know it stands for Wilms tumor 1. It's expressed in many different types of tumors. And at one point, it was actually ranked as the #1 cancer protein with potential to develop a cancer vaccine. And the reason why is because it is recognizable by the immune system. So the first question we had was, how common is WT1 in cholangiocarcinoma? We know it's common in other cancers, but what about cholangio? To address this question, we went to a publicly available database called TCGA, and we simply typed in WT1 in the cholangiocarcinoma group. And what we found, setting a threshold for positivity at around 20% there, is you can see about 20% of cholangios from patients in this database actually express WT1. We then have a collaborator who's a pathologist in Germany, Dr. Diego Calvisi, who helped us out by staining his collection of 156 cholangiocarcinomas And you can see that these were all cholangiocarcinomas because these were staining positively for CK19, both of them. And on the top, you can see an example of cholangiocarcinoma that is positive for WT1, and on the bottom, you can see one that is not positive for WT1. And those results almost perfectly match what we found on the publicly available database on the left side. Almost about 20%, 21% of patients were positive for this. So we think it's a promising biomarker. And I work in a basic science research lab, and we have a number of different mouse models of cholangiocarcinoma. And they're all slightly different from one another based on their molecular biology. So one question we had was, well, how many of them are positive for cholangiocarcinoma? And interestingly, also 1 out of 5, or 20% of them, of the models were positive for cholangiocarcinoma, specifically one that used the oncogenes AKT and a reduction of FBXW7. So that was the model that we selected to look at this, this peptide vaccine in. And here's our confirmation of that. On the top is a Western blot, and you can see that the AKT FBXW7 groups all stain very positively for this protein. And on the left side, another model really does not stain very positively. And it was the same thing when we looked at a microscope slide stained for, again, the WT1 protein from this AKT FBXW7 model. So really the last part of this was essentially the hardest, where we had to predict a WT1 peptide sequence that would actually work in the mouse and cause anti-cholangiocarcinoma immune system to be boosted. So we essentially had another collaborator in Augusta, Georgia. His name is Yukai He, and he's an expert in doing this, in predicting peptide sequences that will boost the immune system specifically. So we asked him to do this for WT1. He took the whole length of the DNA sequence, analyzed it computationally, and he found the #1 most reactive peptide that with potential to be a vaccine that he recommended that we try. So we've right now created both that peptide. We've also created a reagent that will help immunize the mice and prepare them to respond to a WT1 vaccine. And finally, we also generated, with the help of the NIH Tetramer Core facility, a reagent that will help us to detect the WT1 specific T cells. And so now this is sort of where the project is currently. We're inducing cholangiocarcinoma with the WT1 positive model, and we're working on injecting these mice and inducing cholangio either with or without the vaccine to test its efficacy. So in conclusion, immunotherapy is certainly a promising option for patients with cholangiocarcinoma, and WT1 is a potential target. It's a biomarker in about 20% of patients with, this is for intrahepatic cholangiocarcinoma. We know that peptide vaccines can be useful. Unfortunately, none of them are currently FDA approved, but they're a very exciting field of research. And further optimization is currently happening in my lab to prepare to study this WT1 peptide. I just want to thank, first of all, the Cholangiocarcinoma Foundation for this unbelievable event. These are my collaborators. Those are the members of my lab. It was just me, I should say, when I got this grant. And as on a personal note, the Cholangiocarcinoma Foundation grant was my first grant I ever got. It kind of pushed me toward continuing and actually establishing a lab, which I was unsure of at the time. So I just really want to say thank you to Kathleen and Paul Sidenblad for their support, and really just this entire incredible organization. So thank you.
Thank you, everyone. And with that, we'll conclude this session. Thank you to all the sponsors of the Research Fellowship grants and to the awardees for presenting your progress updates. At this time, I would like to ask the presenters, the Fellowship Award presenters, to come up here to where Stacy's sitting at this table up front for pictures and to get your gifts.